But our first speaker is Jessy Carlson, and she is a graduate at the University of Minnesota Duluth and a member of Dr. Julie Ederson's lab team. Cook County is her home, like we've been talking about, so she lives way up north, and her work is directly related to the preservation and health of the forests, and she shares with her neighbors. Jessy previously received a Bachelor of Science degree in biology and First Nations studies from the University of Wisconsin Superior and a Master's of Environmental Studies in Northern Environments and Cultures from Lakehead University at Thunder Bay, Ontario. The title of her presentation tonight is Mycorrhizal Associations and Assisted Forest Migration. So take it away, Jessy.
My name is Jessy Carlson, and I am in Julie Ederson's lab. Yes, and I'm a graduate student at UMD in Duluth. Julie Ederson, she works with population genetics, and our lab looks at the evolution of plant communities as they respond to anthropogenic impacts. In particular, right now we're looking at climate change and how evolution of plant communities is related to climate change. Another one of my close partners that I'm working with a lot is Meredith Cornett from the Nature Conservancy, and she is the climate change director for North Dakota, South Dakota, and Minnesota. There are many, many, many more people involved with this work, but just to give you an idea, those are my partners. So to set the stage, before I start talking about fungi, a little bit about climate change. We all know climate is changing. It's changing a lot more quickly in Minnesota than the global average. And you can see a lot of the most extreme change is in the north of the state. And in the winter, it's warming even more quickly. We are losing our cold winters. So what this means is that our hardiness zones are shifting around. If you ever lived in zone 4 and then moved to zone 3, you would have had to learn to plant different plants in your garden. Same thing is true if the climate moves around. We're probably learning to plant different things in our garden. So what does this mean in a forested context? Well, if we have the climate moving from south to north or from southwest to northeast, we're probably going to see shifting forest composition as well. So this light green here is our eastern broadleaf forest. It's this narrow band of transition between the prairie and the more subboreal forest. This is called the Laurentian mixed forest. We expect that the species that you would find in the eastern broadleaf forest are going to survive and succeed a lot better in the climate that we expect to see in the more northeastern areas. So this should come as no surprise to you. Boreal species are expected to decline, and temperate species would then be expected to increase. However, increases in those temperate species depends on their ability to reach those new areas that are now more conducive to their particular genetic adaptations. And as we all know, trees can't walk, they don't fly. The only way they can move from place to place is by dispersing their seeds. This becomes a problem when there are barriers in the way. This could be mountains or oceans. It could also be cities or superhighways. So if we are concerned about the genetic material from more southern areas or lower elevations, if we're in a montane area, if we are concerned about that genetic material getting to a more a safer place for them to survive, further north or further up a mountain, we need to consider whether they can get there quickly enough and if there are any barriers in the way. Another thing to consider is that if the genetic material that we're hoping will move into a more conducive environment, if that genetic material is killed by the climate before it can disperse, then we lose our seed source for the future forest in the projected climate. So if humans want to do something to help to make sure that we do have a forest in the future, we can do something called assisted migration. So assisted species migration would be if we took genetic material, usually in the form of seeds, from one species and then planted it in a new area. So you're taking one species and introducing it to a new community. But that is not the only way that assisted migration can happen. Within one species, there are many different genetic communities, populations within that species. So normally, the genetic material from one population would spread into a place where it could do really well, and gradually these adaptations would end up where they are best suited for. With the speed that climate change is happening, we're seeing that is not happening. So assisted population migration would be if humans decided to gather seeds from one area, from one genetically unique population within one species, grow those seeds into seedlings and plant them in a different area. And we call that assisted population migration. So does this work? This is a map of the DNR's seed zone. So traditionally, these seed zones worked really great because the state would only gather seeds from the immediate area where they plan to plant, because those seeds would have adapted to that area. The problem is our areas are no longer what they were. So those seed zones contain genetic material that are adapted to what that place used to be.
Recording in progress.
So what Julie's lab has done over the past 10 years is they have taken seeds from zone 105. They've grown them and planted them in zone 104, and then monitored their success and compared them to the trees that were grown from seeds in zone 104. And they found, after 10 years of watching this, that the oaks that were grown from acorns gathered in 105 are performing much better as far as hydrology, phenology, many other factors than the oaks grown from seed gathered in 104. So this gives us reason to believe that this might be a strategy that could be in our toolkit in order to help preserve our future forests. So for a little more about the complexities of this entire approach to climate adaptation, I highly recommended this book. I just finished it. It's a really great book, all about moving plants around. Okay, so if this is what we want to do, if we choose to invest our resources in this sort of work, we have to determine which species we want to invest in. So there are 3 criteria that we want to think about. Does this species have a chance at surviving in the projected climate of the future? Does it have ecological value, and does it have high cultural value? If our resources are limited, we would like to pick species that hit all 3 of those criteria. For my research, I am looking at northern white cedar, Thuya occidentalis, also known as Gijik. And this is the most sacred tree in Anishinaabe worldview. It is also a tree that has high cultural value in non-native communities as well. I'm also looking at paper birch, Wigwasi matig. This is also a high cultural value tree. It also has a shot at surviving in our projected climate scenario, potentially if we help it. And it has high ecological value too, of course. So let's say we do decide that this is a strategy we want to move forward with, and we are planting climate adapted seedlings is what we refer to them as, grown from seed from a warmer climate, and we plant them in a new place. If they don't succeed, we have wasted a tremendous amount of valuable time and resources, and we could risk having big gaps in our forests. So we need to make sure that the way we're doing this, we're doing it in a way that those seedlings have the best chance at survival. So this is where fungi come into the picture. There are many factors that play into whether these seedlings will survive, of course, but I am choosing to look at below ground interactions, specifically mycorrhizal. And as you are all, I'm sure, very well aware, mycorrhizal associations are really important to forest health, to seedling survival and establishment. And we're just learning more and more about that every day. So a little bit about the ecological specialization of mycorrhizal fungi. We can group them into basically functional groups. There are many species of mycorrhizal fungi that would fall into a group that we call ectomycorrhizal, and many fall into a group called arbuscular mycorrhizal. And we'll get into that a little more, but what we need to understand right now is that where we live in our northern environment, most of the forest trees where we live, they host ectomycorrhizal fungi, or EM fungi. Grasslands and more equatorial areas also tend to host arbuscular mycorrhizal fungi, or AM fungi. So in our area, we expect to see EM fungi on the roots of the trees in our forests. And we would expect to see AM fungi in our grasslands and our prairies, or in trees that are in warmer areas. So ectomycorrhizal fungi. This is what a root tip would look like if it's colonized by an EM fungi. And you'll see in the diagram here, the fungal cells, they form a sheath around the root tip, and their hyphae extend out from that sheath, and those hyphae can gather nutrients and water from the environment. These cells also come into the root. They do not go into the cells, they go around the cells, and they form what we call Hartig net. And this is where the interface happens and where the plant can exchange sugars with the fungi, and the fungi can provide nutrients and water to the plant. EM fungi are very important for nitrogen uptake in plants, which is, coincidentally or not, also a very limiting factor in more northern environments. Nitrogen tends to be increasingly more limited the more north you get. EM fungi are also increasingly the type of mycorrhizal fungi that you see the further north you get. A little bit about AM fungi. So these are microscopic, and they actually go inside of the plant cell, inside of the root plant cell. And they form what we call an arbuscule, which is how they get their name, arbuscular mycorrhizal fungi. They also extend the reach of the plant roots in order to access resources that the roots can't reach by gathering minerals and water from further afield than the roots can reach. They can also get into smaller spaces than roots can get. This is true of EM fungi also. So they're gathering the nutrients and the water, delivering them to the plant, and the plant is delivering carbohydrates, sometimes lipids, back to the fungi. So an interesting thing about AM fungi and cedars. Cedars break the rules of the northern forest. Birches host EM fungi, but cedars host AM fungi. So this presents some really interesting research areas, because it's pretty rare for us to have AM hosts in our forests. There's a lot to learn about how these relationships function, which is part of the reason why this research is going to be so interesting. So a little more about AM fungi and their ecological role. So I did say that EM fungi are really important for nitrogen uptake. AM fungi, not so much, although we're learning more. What we do know is that AM fungi are really important for phosphorus uptake, much better at uptaking phosphorus than EM fungi are. And this is the work of Guswarni Anwar. This is her PhD work, actually. She looked at cedars and mycorrhizae And this graph shows cedar seedlings grown here. This is the control. And then she grew cedar seedlings with a mycorrhizal inoculant here. And then she grew seedlings just with fertilizer here. And then these are the seedlings that had mycorrhizal inoculant and fertilizer. And you can see the phosphorus content of each of those seedlings was greatly improved by having the mycorrhizal association present. And this is important because phosphorus is especially important for seedling establishment and survival. So if you want that seedling to survive long enough to leave seedling stage, phosphorus is incredibly important. So if we're concerned about seedling establishment and survival of cedar, our muscular mycorrhizal associations might be playing a really important role. So what are the implications for assisted migration of all of these factors, especially when we're talking about cedar? If we are growing seeds in a greenhouse setting, should we be inoculating them with some sort of mycorrhizal inoculant? Or is it more important that we are looking at the sites where we are planting these seedlings and making sure that those sites have the fungal partners that that tree needs for survival? Or is it a little bit of both? So what I am doing is I'm looking at both. So this fall, I will collect cedar seeds and we will be tracking the genetics of them. So we'll be tracking which trees they came from. And then at each tree where we collect the seeds, we will also collect soil samples. And that's how we will get our fungal inoculant in order to test whether those seeds germinate better in the greenhouse with their native or natal inoculant versus a commercial inoculant. So here you can see a typical growth medium that is pretty popular right now. It says it has Mycorrhizae in it. It has one species of fungi in it. Because I don't have my notes, it is it's an AM fungi, and the species name is Intraradices. So that is what is contained in this pro mix. But it may be that seeds really need the complete community that they came from as far as the fungal community. So we'll be testing that. Right now, this year, because I only started this program in January, so I wasn't able to collect the seeds previous fall, so we do not have a natal fungal inoculant for the seeds that I'm working with right now in our pilot project. However, I am working with NRRI, and they are looking at small mammals as spore dispersers. And they just happen to have a bunch of voles that they have already identified the contents of their guts. They know which fungi, fungal spores are present in these guts. And they want to make sure that the voles that were sacrificed for science get used fully. So I will be given access to that material, and I will be using the scat as a source from my fungal inoculant just for this summer. But again, in next summer, when I'm collecting the seeds that I will actually use for my thesis work, we will definitely be gathering fungal material directly from the trees where we are gathering the seed. Okay, so the other hypothesis is that trees will survive better if we plant them into an existing mycorrhizal network. So it could be that inoculating them in the greenhouse doesn't do much, but being able to have it, giving them the opportunity to plug into an existing functioning mycorrhizal network is the real key to their survival and establishment. So that means we would need to know where those networks are. It could be that we should look for stumps of trees that are hosting the correct fungal partners that we know that our seedlings need and want. It could be that we need to plant them near clearing edges. We need to know how far those networks extend into the clearings where we might be planting. So in order to figure this out, we'll be doing two things. We will be gathering soil samples and doing genetic analysis to determine the DNA of the fungi present in that soil. The only problem with this is that we don't know if all the DNA we're catching is active, basically living, or if it is in a spore and inactive and maybe unviable. So in order to make sure that the DNA that we are identifying in our soil samples is viable, living, active mycelial network material, we're also using what we call an ingrowth bag over here. So what this is, it is a 50 micron nylon mesh that's been folded in half, and we seal the edges and we filled it with sterile sand. We've made 200 of these for this summer. And if this works, we'll be making a whole lot more over the next 3 to 4 years. And what this allows to happen is fungal hyphae can grow through that mesh, but roots cannot. So if there is living mycelium in that soil, it will want to grow into there because it's an empty environment where it can get a lot of nutrients and water. So if we bury those bags in the soil and come back and retrieve them months later, years later, we will be able to determine which taxa of fungi are actively growing through that spot of the soil. So this is what we're doing at the Greenwood site this summer. So the Greenwood burn, as you probably know, a very large area of land was burned pretty dramatically this summer. We have access to that site and we will be looking for fungi in the soil and determining which type of fire kills the mycelial network, which type of fire spares the mycelial networks, or how far from unburned areas does an existing mycelial network or does a mycelial network grow out of. We'll be able to determine how far it grows over the next couple of years into the burned area as well. So what this means for the future of our forests. If we determine that inoculation of seeds and seedlings in nurseries improves their success, then growers should start inoculating their seeds. And we might determine that commercial inoculant works just as well as wild inoculant, or we may not. We may find that the wild inoculant is far better, or that commercial inoculant doesn't really do much. We may also find that it's a matter of site selection that's more important, or that they are both important. And if site selection does turn out to be a factor that greatly contributes to the success of these seedlings, that would mean that before we invest a lot of effort into planting these seedlings, we should know where those networks exist. We should either be able to have some sort of a proxy, for example, existing stumps or certain distances from existing forest edge, or perhaps do some sort of reconnaissance before we do our plantings in order to determine whether we are planting our seeds in a place where they can actually survive. So, summing up, what we know, we do know that assisted population migration does close the gap between trees' rates of genetic adaptation and the rate of climate change. We also know that mycorrhizal associations do improve seedling success. That has already been proven. But what we don't know if inoculation does improve seedling success. the work that's been done on this already has been pretty inconclusive. Sometimes we think that it does seem to help, sometimes it doesn't. It might depend on species. So we need to know more about that. We don't know if the inoculant used in a nursery setting, if the commercial inoculants work as well as a natal inoculant, as well as an inoculant that's derived from that tree's or that seed's natal environment. And we don't know if site selection is more or less important than inoculation. And we don't know if all of the fungal partners that a seed evolved with need to be present in its destination location. So we're not sure if inoculation is more important than seedling planting site selection, or if it's a combination of both. And that is what I have for you today. If you have any questions, I know we have quite a bit of time. I didn't have my notes handy, so I'm sure I missed some really exciting stuff. So please ask me questions.
Did have a question in regard to by moving the tree species, what is the effect on the overall ecosystem, animals, insects, plants, fungi? Do we have any sense for that?
Right. And this is why assisted population migration is a much safer bet, because then we're not introducing new species to an area. What we're doing is taking the exact same species, but gathering seed from an area that has unique genetic adaptations to a slightly warmer, slightly drier, slightly floodier environment. So then you have the same species. It's much less of a risk of creating any kind of environmental upset. When people are moving new species around, that is definitely something to be done with great care. I would say that the book I recommended, it is a book for the general public. It's really easy read. It's just is beautifully written, and it addresses a lot of those concerns, I think, in a really good way. But those are valid concerns. So we're kind of stuck between a rock and a hard place, because if we lose our forests, the effect, the trophic cascade of that would be quite tremendous. When we think about insects, birds, the hydrologic cycle, that would be really extreme. But we also know invasive species can have really extreme trophic impacts as well. So those are valid concerns.
There's a couple more questions in the chat. Ron is asking, what would be the effect of doing nothing?
Right. So that comes back to the potential of large gaps in forest cover. So what that would mean is, a good example would be just looking at hydrology. Trees suck up a tremendous amount of water out of the soil. They move it and hold it on the landscape rather than letting it run off the landscape. They also pump it back up into the atmosphere. Without that water cycling capacity, we would risk losing a lot of soil. There would be increased erosion, a risk of increased erosion, an increased risk of drought, as that water would escape that biome rather than being held there by the vegetation. There would also be the risk of a whole lot of invasive species moving into that newly emptied environment, more ruderal species that can grow pretty much anywhere and take over. So that would be another risk. Yeah, and once again, I mean, not to sing the praises of this book too much, but I just finished it. So I think it addressed all of that really well too. We're in a kind of a tight spot. These aren't easy decisions. And the people who are contemplating doing this understand that this is something that, we have to do it right. We also have to be careful that that we take responsibility for the mess we created as well, and just be careful.
There was another question about the bags that you were capturing the mycorrhizae with, and are they enriched in nutrients to lure the hyphae?
No, they're not. It's just sand. And we sterilized it so that we can hopefully, well, we've eliminated the possibility of, doing a DNA analysis of it and finding a bunch of spores that were in it when we bought it from the Home Depot. So it's sterilized sand. No, basically it's just an environment where the hyphae know that here's an empty environment that isn't already being utilized by other fungi, brand new territory, lots of water, and sand has plenty of mineral nutrients as well that the fungi can break down and harvest. So we don't need to be augmenting that at all.
I had a question about sort of collecting seeds from trees. Does it help to collect seeds from a tree that has weathered a lot of seasons versus a young tree? Like I had some oaks in my yard that are, 150 years old. Have those withstood more than, say, a 20-year-old tree that's dropping acorns? Or are the younger ones more adapted to whatever the newer kind of climate is?
Yeah, that's an interesting question. I have not heard of anyone who has investigated that, so I've definitely not seen any data on it, but that would be an interesting thing to consider. Because like you said, perhaps the younger tree has acquired some adaptation that maybe, allowed it to thrive and survive seedlinghood. Whereas, the older tree might have proven that it has what it takes to survive for a much longer time.
I feel this is different. I feel bad when I'm pulling ash trees out of my garden because I always see them as kind of weeds, but now knowing that a lot of them are dying, it makes me wonder, like, are any of the young ones possibly resistant to, emerald ash borer or something because they're so young and weedy?
Yeah. Well, or you could think about it as you're creating a fire break for the emerald ash borer. Right.
Oh, right.
Yeah. There are people who are saving ash seeds, especially a lot of Native communities. Black ash is incredibly important to them. That for them, there is no replacing that tree. We can't move in another tree that kind of does the same ecological function because to them, it doesn't have the same cultural, spiritual significance as the black ash does. So some people are saving seeds and hoping that once the emerald ash borer moves through and the environment comes back into equilibrium, that maybe we could plant those trees again. Yeah, there's so much more I could say about that. It's a fascinating puzzle.
Mm-hmm.
Mm-hmm.
Gene says he's planting cedars on his land north of Duluth. So far, so good. And then he liked Suzanne Simard's comments on thuya.
Yeah. First of all, make sure you fence for deer if you haven't already done so. What's interesting is, I don't know if you all know of Chell Anderson, but she was involved with the Minnesota Biological Survey for I don't know how many years. It was her entire career. And she has been talking to me about what she saw with cedar and regeneration. And there's not a lot of data on this, but it seems like regeneration rates of upland cedar are maybe at a standstill, whereas lowland cedar sites seem to be doing better. And the National Forest has decided to assume that this is because of pressure from deer eating the cedars, but they haven't actually tested that, which is interesting because, deer go into lowland sites just as often as they go into upland sites. But still, definitely, we need to be excluding deer from our cedar plantations in order to give them the best chance at survival. There's also some evidence that, so it was thought that as climate shifts, as warming moves north, that cedar range would expand further north. But there's some evidence that actually they're getting pinched because they are experiencing drought and flood at the north edge of their range. So just keeping an eye on water availability might also give them a bit of an advantage.
I was asking, I know Jean does a lot of studying up north on the land up there and a lot of observing for years. I don't know if, Jean, do you want to report more on your plantings? See if I can. Are you muted? Oh, let's.
Yeah, it looks like he's talking, but
We're not hearing. Oh, let's see if I can unmute Jean.
How about now? Can you hear me now?
Yes, yes.
I had my earplugs in here. Okay. Yeah, I'm kind of doing the reverse of what you're talking about, Jesse, in terms of the seedlings that I'm planting are coming from up on the Gunflint Trail from a woman who has lots of cedar seedlings growing underneath her house, or her porch. But they do well here, just about 20 miles north of Duluth. And I'm trying to do diversity here. I've got about 20 acres and lots of dead balsam from spruce budworm. So I'm looking to do a diverse forest here. So far, so good on the cedars. They're surviving well, making sure and yes, of course, they're protected. I've got a small fortune invested in seedling cages.
yeah. So you know what you're doing.
Hopefully.
Yeah.
Awesome. Thanks, Jean. It's always nice to hear from you. Ron is asking if politicians are aware of the importance of this issue.
Well, so for a while, I took a little detour in my career away from ecological science and toward more of a social science focus. And that's a much bigger problem, I think, than figuring out what cedars like. So I think I'm going to leave that to people who have thicker skins and tougher hearts than I do. But definitely, like the political process, obviously that's the clincher, right? So I don't know. I don't think I can answer that. I think it would probably depend on the politician, and what they know might be different from what they feel safe saying. Yeah.
Any other questions for Jessy?
There's a question here about epigenetics,
Oh,
Oh, yeah.
which I'm not familiar with that. It says, I'm not sure how epigenetics works in trees, but an older tree be able to pass on life history specific epigenetic benefits to its seeds.
Oh,
Yeah, I think that's from Charles. I think he was commenting, weighing in on my question about saving seeds from an older tree versus a younger tree.
yeah.
Yeah,
I
that
have
was
no-
just a comment. Oh, sorry.
Oh, that was you, Charles, who commented on that. I have no idea if anyone has even looked into that. Like epigenetics is such a, I mean, not brand new, but it's kind of a new-ish thing in human genetic research, right?
Yeah.
Yeah, so anything that would be plant related would be somewhat behind where we're at with understanding human genetics. So I wouldn't be surprised if no one has formally investigated how epigenetics, I would assume epigenetics happens in trees. Why wouldn't it? But I don't know if anyone's looked into it.
The thing that made me think about it was we had, again, an old tree that I hated to lose, but I watched it for years and had an arborist come in and they said, yeah, you can't save it. But the guy said, but you could plant a bunch of the acorns because I bet this is old enough that it might survive differently than some of the new stuff that's available out that people are buying and planting. And it just, the arborist sort of was interested in, because it was a white oak and it was so large that he felt like its offspring would be interesting to him versus the white oaks that you're buying at a landscape store.
Absolutely. There's someone in Julie's lab, an undergrad actually, who her undergraduate research was looking at native seeds of a particular wildflower that is grown very responsibly by native seed company. But she determined from her research that there was artificial selection happening because of the processes that we use in order to harvest the seeds that we are growing. So there was actually a loss of genetic diversity in these seeds because of the design of the equipment and which seeds made it all the way through the process and which seeds were either too big or too small. So then you end up with a genetic bottlenecking of the seed lot that ends up being sold and planted. Whereas in the wild plant community, that genetic diversity is much greater. And as we all know, genetic diversity is how living beings hedge their bets for unexpected upsets. So that genetic diversity is incredibly important for resilience, especially as we move into a more unpredictable and potentially dangerous future for our plant communities. Yeah, diversity is always a good bet. Which is one of the reasons, I'm focusing on the mycorrhizal community, but Dr. Edison's work is definitely about that genetic diversity and making sure that when we are gathering seeds for reforestation projects, that we're not gathering seeds just from one tree, that we're gathering seeds from many, many, many different trees that are representative of many, many, many different genotypes, so that we maintain as much genetic diversity as we can, because we wouldn't want to accidentally swamp an entire region with the genetic material of one tree. That could be dangerous. It would reduce our resilience. The genetic diversity limitation would reduce the resilience of those communities in the future.
It's also fascinating. Thank you so much. This has been amazing. We're really proud to help sponsor your work.
Yeah, thank you so much.
Well, and then Charles, who we were just chatting with, is our second speaker. Charles obtained his BS in biology from Earlham College. Afterwards, he worked for 5 years as a medical technologist at the University of Minnesota Veterinary Medical Center, while pursuing a Master of Biology through the College of Continuing and Professional Studies. In 2021, he transferred to the Bioproducts and Biosystems Science, Engineering and Management graduate degree program. He currently works in the Zhang lab, pursuing the lab's primary goals of developing a brown rot fungi gene editing system and harnessing fungi for remediation of environmental pollutants. The aim of his research is to leverage the Zhang lab's experience in fungal genetics to study the genetic underpinnings of fungal metabolism and recalcitrant xenobiotics, namely PFAS, per and polyfluoroalkyl substances. I'm sure you're good at saying that much faster than I am, Charles.
It's a mouthful.
To elucidate the mechanisms through which these compounds can be degraded and move toward the development of microbial treatment for these emerging contaminants of concern. So the title of Charles's lecture to us tonight is Mycoremediation of Forever Chemicals. So this is really about using fungi to break down the plastics and stuff that have those billion-year lives.
As is just introduced, I'm going to present to you guys on the mycoremediation of per and polyfluoral alkyl substances, which I'm going to abbreviate as PFAS or PFAS going forward. There we go. To start, I will introduce these chemicals. As you've been told, they're known as forever chemicals because they last so long in the environment. Unfortunately, they're also environmentally ubiquitous, and that's because there is over 8,000 of them, depending on how you decide to classify the chemical compounds, whether or not you actually consider it a PFAS or not. These compounds are truly ubiquitous. You can find them almost anywhere in the environment, anywhere you care to look. And that's because they are in just about everything. They're used a ton in industry, but for our purposes, generally anything that is waterproof, stain resistant, grease resistant, etc., probably has or has had PFAS compounds in them. So this is anything from, your waterproof rain jackets, or the big one I bet we're all familiar with is Teflon. They make it with a different compound nowadays, and it's up in the air whether or not it's any better. But that was PFOA in the past. And so you can see sort of the general categories that you may run into them in your day-to-day lives. And then this crazy chemical compound here, this is PFOA. And this is just a good idea of the general structure of these compounds. They are per or polyfluorinated. So this just means that it's a fluoroalkyl chain where normally these would just be hydrogens and it'd be a pretty standard compound, but they've all the hydrogen atoms have been replaced with fluorine, which gives them their properties, but also makes them a real pain in the butt if you're trying to get rid of them. So this is a beast of a figure, but this is sort of the phylogenetic tree of PFAS, not inclusive, and they have a million different names, a bunch of subdivisions within them. But I want to focus here on this red box because it's the perfluorocarboxylic acids, PFCAs, and then the perfluorosulfonic acids down here that are my main focus. So there's PFOS and PFOA are the two compounds I work with. And this is a pretty cool figure. It shows all of the articles printed. This is a few years out of date now. So the numbers have changed, but as you can see, PFOS and PFOA have the most work done on them. And for good reason. They're considered legacy PFAS. They've been around the longest, and they're also the ones you find everywhere. Not to say that you don't find these other compounds too, but in almost any environment you find PFAS, you find PFOA or PFOS. And I'll give a little bit more information on why that could be coming up later. So as mentioned before, I focus on the legacy PFAS. And then here they are. Luckily, they've largely been phased out, and there's new legislations and regulations again banning their use in many cases. However, it's really sporadic. Like I say, they're banned here in America and a few other places, but they still get used a lot elsewhere. And even here, they are still used for certain things, simply because they really are very industrially useful. And so maybe they've been phased out of like fast food packaging, that sort of thing. But there are some jobs that these compounds do that they're really just the best of the best. And so it's difficult to switch them out for anything else. And just a quick couple fun examples. These things since they're really resilient, they make really good seals in this industry. So at a space station, you probably don't want to use a second-rate compound. So it's like stuff like that, where it's still very hard to phase them out just because they're so good at what they do. And that's being I'll also mention that, unfortunately, in the environment, PFAS is not quite as inert as we originally thought. And this could be a reason we see so much of the PFOA and PFOS. And that's because when other PFAS compounds are abiotically or biotically changed, they often end up being transformed into the PFOS and PFOA, where they just kind of stay. So if a PFAS compound can be interacted with a microbe, oftentimes they end up as a perfluorocarboxylic acid like PFOA, and very little degradation occurs after that point. So I mentioned the legacy chemicals, but I do want to mention some of the replacements, because these are also becoming important. They've not been as studied before. And if you're listening in the news, I've heard seen a lot of articles, seen a lot of news stories talking about PFAS. So some of these names might become more popular in the media going forward. So you may have heard of Gen X chemicals. Gen X is a large category. They're the PFOA replacements, and that's this massive molecule here. And essentially, the industry has liked to say that these are better, less toxic, not as bad in the environment. And that's primarily because they've broken up that carbon-fluorine chain. They have this oxygen bond here that theoretically could be easier to interact with. However, the little investigations that have gone into these compounds have found that both these two classes, the fluorotelomere alcohols and the Gen X chemicals, are at least as dangerous as the other PFAS. So it's sort of corporate rebranding more than anything else, unfortunately. And then since I showed them, I'll go over them a little bit. These fluorotelomere alcohols, and this one is 6,2-FTOH. These are sort of the precursor compounds. So when they're producing these things in industry, they often start with a fluorotelomere alcohol, and they combine those in various ways to create all of the other PFAS molecules. So they're also super common because they're sort of the base level building blocks of other perfluorinated compounds. So, why are they such a problem? It's because they're water soluble and they can spread throughout the environment. I mentioned they're water soluble. Most of the work that's gone into PFAS has gone into their investigating their fate when they hit the aquatic environments, because been focused on human health. You don't want to get into your drinking water. And so as you can tell from this figure, there's a big focus on PFAS in water, which is definitely important. But as you might also notice, some of these source sites, such as landfills and military bases and airports, a lot of these sources are actually terrestrial in nature. So that means a lot of these compounds are coming from terrestrial environments, not necessarily aquatic ones. So PFAS is also a concern in terrestrial environments. So more on that. So soils as a PFAS reservoir. This is why I'm interested in fungi. This is a nice figure here. It's taken from a really good review. They've normalized the sample depth because they've actually, it's a meta-analysis where they've pulled data from a bunch of different studies. So essentially, the one up here, this is surface soil. So, if you reach down and touch the ground, that would be what they're meaning by that. And it goes all the way down to the water table. So obviously the depth of soil to the water table varies from environment to environment, from area to area. So they've just sort of normalized it so they could compare all this data. And as you can see, there is a significant amount of these long chain, which is PFOA. So that's a carbon chain in these compounds that's 7 or more. That's how they've classified them here. Depending on where you read, it might be greater than 5, greater than 6, but the long chain ones tend to stick in surface soils, which makes sense. They're bigger, they're longer, they're a bit more bulky, they grasp onto things. But that means that surface soils, where all of the microbial life is, are exposed to these. And what's more alarming is, while there's always been a focus on PFAS in water, it's found that the concentrations of PFAS in the soil tend to be higher. In a water, it might be parts per billion. In the soils, it can be parts per million. So it's like an order of magnitude greater concentration in the soil, which is going to be a problem because you can treat water all day if more is just going to keep flowing from terrestrial environments into aquatic ones, then you're not really attacking the true problem there. So I want to bring in a local story and why I'm particularly interested in this. And that's because PFAS is a large problem in Minnesota. It's a large problem all over the place, but unfortunately, perhaps Minnesota might be emblematic of PFAS pollution. So I'll start with this figure on the right. This is and looking at some polluted groundwater and municipal wells. So these purple areas, this is Woodbury, by the way, it's almost my backyard. You can see that a large amount of these wells are actually contaminated with these compounds. And this is because of 3M manufacturing facility. 3M actually they no longer do as much, but they produced a large amount of these PFAS chemicals for a very long time. And as you can see, they've escaped into wells. So this gray area is a source. I'm not entirely sure where this is, to be honest, but this, I believe, is actually a 3M facility. If you're to zoom in here on Google Maps, this would be a 3M site. But then the Minnesota Pollution Control Agency, knowing that PFAS is a problem in our waste streams, simply because they're in so many products and so many of the things we make end up in our garbage, they knew that there was likely PFAS contamination in a lot of our landfills. So they did a survey of just over 100 now closed landfills, sort of like legacy landfills, and they found that in 98 of them, so essentially all of the ones they cared to look at, they found excessive levels of these PFAS chemicals. And so this is what this is, and you can tell, so all over the state, there's impacted wells, and unsurprisingly, the ones closer to our neighbor or my neighborhood, where 3M was located, where they're producing a lot of these chemicals, there's quite a large amount of these landfills where there's PFAS contamination 10 times greater than the EPA's advisory limit, which, forgive me for forgetting at the moment. So there is a lot of PFAS in our soils and in our landfills. So why are they so recalcitrant? Why are these things going away? I could say a lot about this, but then for time, maybe I'll skip over a little bit of it, but essentially it poses a problem for microbial metabolism, and they are highly resistant to any sort of abiotic influence as well. And this comes down essentially to the strength of the carbon-fluorine bond. It's one of the strongest chemical bonds. Some say it's the strongest chemical bond out there. And if you look at these compounds, they have a lot of carbon-fluorine bonds. So these do not break very easily. And they also pose a problem for microbial metabolism because they're so hard to break, it's nearly impossible to gain energy from breaking them down. So not only are they toxic, they're not good food either. And so most organisms don't interact with them because it'd be a negative outcome if they did. And so I just wanted to compare and contrast a man-made fluorinated compound, PFOA, with sodium fluoroacetate, which is one of the rare examples of a naturally occurring fluorinated chemical. And as you can tell, they have almost nothing in common. Natural fluorinated compounds, when they do exist, have a single fluorine bond. And a single fluorine bond, as hard as that is to break, is an order of magnitude easier to break than multiple fluorine bonds. They actually have an effect where multiple fluorine bonds tend to strengthen each other due to just the contortion and the shape it puts on the actual molecule. So the more fluorine you have, the harder it is to get rid of that fluorine. So, which is why they designed them this way, because the chemists or the industrial chemists knew that, and so they designed them this way to make them as recalcitrant as possible. So where do fungi come in? Can fungi be able to do anything about this. We've known that you can use fungi for other things, such as the polycyclic hydrocarbons or PCBs, polychlorinated biphenyls, another halogenated man-made molecule that it was a problem. So there's two ways. There's mycoremediation, essentially using fungal metabolism to break these things down, or even a relatively new idea of mycofiltration. Can they be used sort of as a filter to remove them? And that's what I'm investigating. So I've gotten into the habit of really trying to justify using fungi for these purposes because most of the focus tends to be on bacteria. But as I said, soil is a significant source of PFAS contamination, and the majority of soil microbial mass is fungal in nature. So how fungi interact with these compounds is going to be important for their overall environmental fate. And there's also been some good evidence that fungal enzymes can interact with these compounds. So perhaps the fungi themselves can, even though there's been very limited work in that vein. Another pretty cool thing, which I think I'll just get into a little later because I've got some data on that. But so once again, three key traits of really why I've chosen fungi. One is this large hyphosphere area. So that's just the area of their hyphae, the area around them. There could be literally miles of fungal hyphae in the soil. So that means they can infiltrate the soil, spread throughout the environment, and potentially remediate large areas of land. Also, if you were to use a lignocellulose growth substrate like wood chips, which is what I'm investigating, this would give them a solid foundation for continued growth, because in any bioremediation approach using a live organism, the stability of your microbial inoculant is always of a huge concern. If you're going to introduce a bacteria or a fungi into an environment and hope it grows and breaks something down, that might not always work because there is a resident microbial community. And so your organism is going to need to compete with what's already there. And oftentimes it doesn't work too well. And also, fungi can perhaps avoid some of the downsides that bacteria face when they interact with these compounds. And if anybody has more questions on that, I could point you towards a really good review article of just why bacteria have such a problem with these compounds. And I could explain a little bit more too, but I'll skip it for now. But fungi, most of their degradation can occur extracellularly because they're osmotrophs. They excrete a huge quantity of these extracellular enzymes that break down the things in their environment so that they can then suck up all the nutrients they release. And so here in this figure, this is just a fungal lacase. I believe this one is from Trametes versicolor, showing it acting on both lignocellulose and then these other blocks here representing xenobiotics, or just other compounds in the environment, because these enzymes are broadly specific. So my research questions: how do fungi interact with these compounds? Do they degrade them, or do they perhaps sequester them? Maybe they absorb them, maybe they just bind to their hyphae. And if so, what are the biological pathways involved? Because that's sort of a black box right now. Even though there's some evidence fungi do degrade these compounds and they are interacting with them, precisely how is unknown. And then, can these fungal PFAS interactions actually be used for some sort of mycoremediation application? So these are some relatively arcane looking figures, and they're kind of beastly, but I just wanted to highlight here that in these proposed microbial degradation pathways for PFOA, you often find that, or for PFAS, you often find that PFOA or PFOS is actually sort of the endpoint. So this has been some work done on by microbial consortium from a wastewater treatment plant or even a landfill. Often shows you feed microbes a PFAS molecule, and what comes out is PFOA or PFOS or another perfluoral carboxylic acid, which are really the most hard to treat. They are the most resilient and the hardest to get rid of. And so when microbes do interact with them, they tend to create a more troublesome compound. However, there is some good work here that suggests that fungi, when they interact with these compounds, can actually shunt things away from these perfluoral carboxylic acids to some fluorinated acids, like this 5,3 acid here. You can kind of tell in this figure here, there is essentially a gap in this fluorinated chain. So these compounds are considered less recalcitrant. I'm not saying they're not detoxified or not bad. They are just empirically easier, if still very difficult to get rid of, than some of the bacterial byproducts of PFAS metabolism. So if there's going to be a microbe used to degrade these things or interact with them, there's some good evidence suggesting that fungi are a good choice. So this is just some more information on lactase, because lactase has been studied in isolation and actually found to defluorinate these compounds, which is really exciting. But in the domain of time, I'm going to skip forward. So this is just a really good review. Once again, lactase itself, not necessarily the fungi making the lactase, but this lactase enzyme that the fungi secrete has been looked at quite intensely, and it's found to actually be able, not entirely to get rid of PFOA, but at the very least reduce it up and release up to 28% degradation and 28% defluorination, which is really what you're looking for is defluorination. You have to break those carbon fluorine bonds. Otherwise, you're not really getting rid of the problem. You can change it into other forms, but it's still a PFAS. It's still recalcitrant, and it's still a problem. Once again, some really arcane figures. If anyone has any questions, I can go through this, but otherwise, feel free to ask me. I'm going to skip through this because the end story here is that lactase is able to break these carbon fluorine bonds. So now I'll get into a bit more of my data, because this is actually kind of exciting. I apologize for the figure. I realize it might be a little hard to see. It didn't translate over into slides all that great. But I started by just seeing if fungi are tolerant of these things. I chose primarily wood decay fungi because that's what I work on. And some of you may recognize some of these compounds. So there's the shiitake mushroom here, Trametes versicolor, schizophilum, foamies. You get to see a lot of that, gliophyllum or asterium. And what's kind of exciting here is that even at a really high PFAS concentration, 500 milligrams a liter, that is truly astronomical. That's an order of magnitude higher than you'd ever expect to see, even in the worst contaminated areas. And they all grew pretty well. On the plates, they grew healthily. Sure, their growth rates are a little slower. You can tell maybe they weren't quite as happy if they weren't growing on PFAS, but they grew on it just fine. And if you compared their growth rates to just regular PFOA-free media, you didn't see a stunningly huge decrease in their growth, which is a good clue that maybe things are fungi are good candidates for this sort of work. Next, since I knew from the literature that lactase had been investigated, I wanted to see, are my fungi producing lactase on PFOA-containing media? And they are. You can see some of them, not too much, but I grew some of the best performing from the PFAS tolerance tests in either glucose or wood, and to see just what their enzyme activity was. And that's what you can see here using this ABTS assay. ABTS is just like a dye compound that when lactase breaks it down, it produces this really brilliant green color. So this is what it looks like on an agar plate with ABTS in it when these fungi grow. And this is a Mycorrhizal plate showing what it looks like in liquid. And so you can just sort of quantify that enzyme expression by the degree of color change. So I then did something which I think is actually kind of cool. This is pretty exciting. I grew fungi in liquid culture, and I actually was able to detect fluoride release from these compounds. Granted, it's parts per billion, so it's very low. But as far as I know, no one has ever actually been able to achieve this. They've never seen actual fungal defluorination of these compounds. And so this just here is sort of the best some of the best performers. And I have a bit more data here. These guys, really excited to see that this Sterium is one of my best performing organisms, even though in some of the replicates, they just died, which I believe has more to do with the liquid culture. These are all wood decay fungi. They don't like growing in liquid. My lab has a lot of experience growing them in liquid, so we can do it, but they definitely prefer solid media. So in the future, we'll be growing them on a solid substrate. It's just that all of these fluorine analysis, since historically we've been interested, or humans in general have been interested in fluorine levels in water, because we fluorinate our drinking water and such. Most of the methods used to detect fluoride perform best in water. And so we started with liquid cultures just to aid downstream analysis of our samples initially. But now that we've sort of done an initial screen and we can really focus in on some best performing species, we plan to expand that to solid media. So this is just a general overview of my project. I plan to take environmental samples, not just using some of the samples we have in our lab here. Gaining access to some of these contaminated areas has not been too easy. I hope to make some inroads there in the future, but some of the most contaminated areas, which are the ones you'd want to screen for any sort of resident PFAS degrading or PFAS tolerant microbe, tend to be on Air Force bases or airports, or some company property, someplace that generally they don't let mycologists walk around on. So I'll then investigate more of the degradative pathway. That's using the Zhang's lab, like sort of resident fungal geneticist and fungal experts there, because we'll be able to actually investigate what genes are being expressed, what enzymes are being produced, and pairing up with the NRRI, who is a great analytic pipeline for PFAS degradation products, we can sort of see what the fungi are doing while seeing what's happening to the PFOA. And those two bits of information coupled together, you can begin to build a more accurate degradation pathway and actually get under the hood and see what's going on here. Simultaneously working with another lab at the U, Roger Ruan, who has a great pyrolysis facility. We plan to take, since we're growing them on solid substrate, these wood chips, we are going to end up with PFOA contaminated wood chips. And we want to see if we can't get rid of that. And so we're going to pyrolyze it and create biochar with it. And biochar is sort of activated carbon, can actually be used to be introduced into the environment to bind pollutants, including PFOAs. So we can both study the degradation of PFOA and also produce a byproduct that could be used in a downstream application to perhaps help clean an environment with it, while hopefully producing some syngas, which could be a sort of biofuel. So some quick future prospects here for this biological treatment. One, I plan to identify some additional fungi. I'd really like to find some environmental isolates, because going to PFAS polluted environments and seeing what's there seems like that might have a higher chance to find something that's both tolerant, able to interact with these compounds, rather than just screening our resident fungal library. And as I mentioned before, I really want to identify what genes and enzymes are being expressed here, so you can figure out what's actually going on instead of just sort of observing the phenotype. You can observe the genotype. And then once you have that information, you'll be better prepared to develop some sort of remediation approach. Unfortunately, it's probably have to be what's called a treatment train, which means you need to combine multiple potential remediation strategies, like the fungi, the biochar, the pyrolysis, or maybe even some other forms of remediation, because these things are really hard to get rid of. And that's the real challenge right now, is nothing in our toolkit is sufficient in and of itself. And so you're going to need to find combinations of things that together can get the job done. But this all comes with some challenges. One, you need a better screening method. What's something I'm working on currently is developing a higher throughput method for screening fungi and microorganisms in general for these sort of reactions. Also, tracking the transformation or degradation products is very challenging, because PFAS is such a diverse group of chemicals. So many of them are sort of their exact formulations or company secrets. And there's very few like actually control solutions. So if you're using the instruments to try to detect these, you often run into the problem that you don't know what you're looking at. So you don't know, because there isn't a nice database or a library. So we don't even, there's so many of these compounds, we don't even know how to ID most of them. And there's so much PFAS used in laboratory equipment that often contamination is an issue. So that's a huge problem. And as I mentioned, the degradation tends to be slow, tends to be incomplete. So you need multiple potential options to actually get the job done. And as far as the fungi grow, we really need to figure out what their optimal growth conditions are. Because as I showed you, even in my best performers, some of my replicates just didn't work. So clearly there's something going on there. There's some conditions that need to be set to make this better. And then perhaps the largest challenge is actually getting any sort of approach we might come up with from the lab, off the bench top, into the environment, into an actual scalable application that can be applied out in the real world. And in any sort of research, that is probably challenge number one. But I think that's all my time. So I'll end with some acknowledgements. I'd like to address the M Drive. They have been really important in funding some of my work. I am working with the NRI. I really wouldn't be able to investigate those degradation products by myself. And of course, the Bioproducts and Biosystems Engineering Department at the U, which I'm from. And then, of course, the Mycological Society. Thank you. I've already used some of your funding for this work, and it's been a real honor for coming out here and presenting. And then I also want to give a shout out to my advisor and principal investigator, Dr. Jiwei Zhang, and his postdoctoral assistant, Li Weiren. The two of them together have been an immense help to me, and the Zhang Lab group and all of us together has really been great. So yeah, thank you. I believe that's all. And if you guys want to read something, I got some references here.
Awesome. Awesome. We already have some questions. Gene says that he spent at least a year in undergrad lost in biochem, including polymer chemistry. So you guys are in good company, and
Indeed.
Still found your way to mushrooms.
Yep.
Ron's asking, what are the harmful effects of PFAS to organisms?
Oh, so I knew I'd get that question because I did kind of skip over that. They are toxic and bioaccumulative. I'll just give some broad categories of ways they affect, because it's one of those things where, once again, it's such a diverse group that simply the research hasn't been done to accurately quantify and qualify the exact health effects of, like, any given PFAS. Generally, though, they've been tied, like so many of the things we make and spew into the environment, with increased cancer rates. PFOA in particular has been linked to a variety of cancers, testicular, breast, thyroid, like you name it, just cancers out the wazoo. They are hormone dysregulators. They really affect, as I said, they cause thyroid cancer. They dysregulate thyroid hormones. They disrupt cholesterol regulation in your body. It actually can cause high cholesterol. And if you actually think biochemically, that kind of makes sense. These are amphiphilic molecules, meaning they have a hydrophobic, that carbon fluorine chain is hydrophobic. And then their little functional group head, that's hydrophilic. And so they've got one end that can interact with, like, cholesterol, which is also similar in your body. So they can sort of bind to the same things cholesterol does, like in your blood. And so it can disrupt cholesterol transport through your body. It causes reduced fertility and low and decreased, like, birth weight. Now, these are all human effects. There's also been some work done showing that PFAS in the environment decreases alpha and beta diversity of the environment. So it essentially kills off certain organisms and dysregulates the environment, as you might suspect. And I haven't gotten too much into this literature, but with COVID and the pandemic, there's been some recent work tying higher levels of PFAS in the blood to increase mortality from COVID and more severe illness. So there's that too.
Man. There's a question from Gabrielle, and she says, just spitballing here, are there ways to screen for lacase or other defluorinating enzyme production through environmental sampling and sequencing and searching for certain genes? Would those genes even be conserved in a degree across different taxas, or for that to be feasible? Or do you have to basically assay in vitro?
So it's a little of both. You can totally screen for that, actually. Lacase, I focus on fungal lacases. Bacteria also produce them too. Generally, I think fungal lacases are the primary drivers, just because fungi tend to produce so many of these enzymes. But yes, you can actually screen for that, sort of like a metagenomics approach. However, you will probably have to follow that up with some actually in vitro assays, just because you can find those genes out there in the environment. Are they being expressed? Are they actually producing that enzyme? Because you can screen for the capability, but it's difficult to use that sort of environmental screening to actually confirm expression. So yes, if you're just going to sample, so RNA-seq, that's actually what we'll be using in my lab to see what our fungi are doing. You can totally, but to do that, you probably want to do that on individual strains, or at least a reduced number of strains. RNA-seq gets to be kind of a bear and I'm not actually sure how you could apply that to something like a soil environment. You would probably want to, at least at the outset, limit the amount of species you're looking with, just so that your end data set isn't just huge. But you could totally actually also do that as well. So you would have to do RNA-seq, but that's a bit more complicated than sort of like a metagenomics approach. It's like, is the lacase gene there, or are these potential other enzymes like peroxidases that could be involved? Are they out there in the environment? And I'd suspect that the answer to that question would generally be yes, because especially in the soil, all these enzymes I'm looking at, lacase particularly, is actually really important in the formation of soil. So it's produces humic substances like humus, that really nice soil. Like as things get broken down and turned into soil, lacases and related enzymes are really some of the primary drivers of that. So anywhere you've got soil, you probably have some of these enzymes.
Wow. I have a question. It's not super scientific, but I know we've heard other lectures about like when you're testing the breakdown of wood from a fungi, that sometimes they do it in succession. Like one is super good at one thing, and then another one's super good at the next thing. Mm-hmm. I don't know if it would be the same in soil, or if you could find out that, in these PFAS compounds, if there are certain ones that are great at the beginning part of it, and then other ones would be able to kind of take over and finish it up as other ones have started. Like that maybe the stronger ones aren't as good at the first stage, but maybe they have their own kind of niche stage that after another one has broken it down, that they could do their job better.
That's an excellent point, and that's very true. And that's one of the reasons I want to do some environmental sampling, because I've been focusing on single strains of fungi, primarily because initially that's a bit easier to investigate. But as I mentioned, like those treatment train approach where you have to use multiple things, if there is going to be some sort of mycoremediation or bioremediation approach using actual live microbes, it's going to almost I would say it has to be a consortium. I think the chances of a single organism being effective enough at that job are slim to none. And so they're going to need some teammates, I think. So yeah, there's going to be a successional process. There's probably some that simply can't do that initial degradation just because those full compounds they're really beastly. And so it's probably a limited amount of organisms can actually take that job on. But that's one of the issues with bacteria, especially. But if you can start snipping those down and sort of put some kinks in their armor, so to speak, some other guys might be able to come in and take advantage of that. And so in the future, I think it's going to have to be more than one organism, like a whole. So But people have done screening on this, like in wastewater treatment plants, because they've been interested. A lot of the water soluble, so a lot of these PFAS enter into our wastewater treatment system, and so they're wondering, does our sort of standard wastewater treatment process, which uses a lot of biological agents, or like active microbes in the treating the sludge, do they actually work? Do they actually do anything? The unfortunate answer is no. They haven't had much luck, but I think that's because most of our wastewater treatment is anaerobic, and the anaerobic community doesn't seem to be able to interact with these that well. So the fungi, the wood decay fungi, their degradation is aerobic, and so they likely would not be present in the wastewater treatment plant, like this lactase enzyme that's been found to break down these compounds, at least to some extent, requires oxygen as a cofactor. So it's actually an aerobic enzyme. So there wouldn't be very much lactase in a wastewater treatment system. Not to say that I think lactase isn't the sole causative agent here. I've just been focusing on that because that's where the research has been, and that's sort of where the information is currently. But I fully believe that there's multiple things at play. In fact, you go back to some, if I were to go back to some of my data, which maybe I just quickly will here, because I think it's actually kind of cool. All of these organisms, for the most part, are white rot organisms. That means they use enzymes like lactase to do their degradation process. But this Gliophyllum trabeum here, the WT just means wild type. So these are a brown rot organism. They lack all of the enzymes that have been researched or may have been suspected to be involved in PFAS degradation. They don't have them. They can't express them. Yet it's still one of the better performing species. It performs better than some of these white rotters that have those enzymes. So clearly there's something else at play here.
So interesting. So we're probably just drinking tons of chemicals when we drink our water. Yeah. Yeah.
Well, I don't.
Yes.
Perhaps I could. Yes, probably. I don't want to scare anyone. I
Wow,
can't
this
speak to
is
that directly.
fascinating.
I will say that, like I showed those figures where there's the contaminated wells, the 3M and has paid to install a lot of filtration systems on some residential wells and waters is to actually filter it out. So I don't know what we're doing with PFAS in our wastewater. So the fluorine in the syngas, I think the fluorine would be removed. It probably, fluorine in and of itself, that's a good question. We haven't actually looked at this yet, and that's something I was wondering too. Likely it shouldn't be an issue. I say likely just because fluoride, once it's released into the environment, it can cause a real problem intercellularly. I mean, that's why we fluorinate our drinking water. It's bad for microbes because it can kind of cross their cell wall. And once a fluoride ion is inside a bacteria, it turns into hydrofluoric acid, and then it messes up their metabolism. And so that's how it slows their growth. But fluorine is really, really rare in the environment. And the reason it's really, really rare out in the environment is because it gets bound to something almost immediately. It gets bound up in minerals. And so releasing fluorine to the atmosphere or to the environment in the form of like this syngas, it's not going to be toxic to us. It's not going to be toxic to any macroorganism, and it's going to find something out there in the world to bind onto and become inert almost immediately. It's really only an issue in the cytosol, in an intracellular environment, in sort of really small micro scales. And so, but yes, the actual pyrolyzing of it should be able to break those carbon fluorine bonds and therefore completely degrade the PFAS compound. Or so our collaborator in the Rouen lab assures me.
I have a pretty far out there question. So lactase is a really good candidate, and there's a lot of research there currently. And I know, I'm not a super strong chemist, but I'm into biology a lot. And I've been looking into like synthetic biology and biotech and stuff. And I'm wondering, is there any research, or maybe a better question would be, how science fiction is it to think about like bioprospecting certain enzymes or variations thereof that are like really effective at defluorinating, or maybe even like Frankensteining and designing a defluorinating enzyme that you could then like mass produce in a vat of E. coli?
I think that's probably, I've been talking up the fungi here a little bit, but I think in the future, that's what people are going to be focusing on. There's a whole field of enzyme engineering. I believe it was 2018, Dr. Sue Arnold, who really sort of revolutionized our ability to do enzyme engineering, has a really cool quote that I'll probably butcher, but essentially she talks about climbing out of a valley to the peak of a mountain. And all you need is like one handhold, because once you have one handhold, you can find another. And that's her iterative process of taking, like, screening, bioprospecting, find an enzyme that can even begin to do the job. Once you find something that can even start, it's fully within our capabilities to optimize that, to make the minute changes, to study how it does and to improve it. And so I think bioprospecting for a defluorinating enzyme and then seeing if we can improve it, that's going to be the way of the future and is well within our potential. And the issue is when people are doing the initial work, because there's a bunch of dehalogenase enzymes, fluorine, bromine, chlorine, these are all halogens. So there's a whole host of enzymes that can break halogen bonds. And so people found the closest thing to fluorine is chlorine. So people found a bunch of enzymes that break chlorine bonds, and they threw them at PFAS to see if they could do anything. And unfortunately, they couldn't. So for a while there just wasn't any of sort of the top candidates that anyone just might think of off the top of my head. Like, well, if you want to break a halogen bond, look at these whole class of enzymes that break halogen bonds. They kind of found that none of them really did the job. But yeah, bioprospecting for an enzyme that can do the job, and then seeing if you can increase how well it does that job, is probably going to be the way of the future. Because ideally, I think someone would like to have an enzyme and just be able to dump that enzyme into the environment. One potential upside of using an actual live organism is you might have increased residence times in the environment, so that they might be constitutively expressing this over time, because all enzymes are going to eventually break down. They're going to lose their activity. And so you're going to see decreasing results over time. To reimply something, the costs goes up. And that
was a really good answer. Thank you so much. I'm going to look into the field of enzyme
It's
design.
really very, very cool. I'm more of a biologist myself. If I was more of a biochemist, I would really love to be an enzyme engineer. Oh, and I see we got another question here in the chat. Where they source the fluorine for when they're producing this, I'm not actually sure how they, where their base level building blocks come from. That's a interesting question that I don't have a good answer for. But is it fluorite? They might be able to find. All right. So does thank you. I guess once again, thank everybody. I don't know if there's any other last follow-up questions.
Thank you very much.
Yeah, this is
Thank you so
Great.
much.
Both
So
of you,
eye-opening,
great
both of these.
presentations. Oh,
yeah.
very, very good. Very, very good.
This is a lot of fun. Sorry, I went kind of over time.
No, our meeting goes till 9. This is perfect. You guys were both perfect and eye-opening and just enough over our heads to make us want to learn more. It's all perfect. Yeah.
Oh yeah, stop share. My bad. Sorry about that.
All right. Any other questions from anyone? I feel like our minds are all full and spinning in such a good way.
So if I work at airports a lot, and we see the PFAS as just being a huge problem. And it's a weird problem because the federal government mandated the use of it for so long. And now like there's no, like you can go after a 3M, but it's tough at the airport. So everybody's trying to get geared up for it. But like how far out is the window of hope of seeing some practical application for this?
Of this specific approach, it might be distant in the horizon. I think, unfortunately, simply because as far as microbial interactions with these compounds go, it's a relatively new field. And I did a pretty extensive literature search, and I was able to find a total of 6 research articles talking about fungal PFAS interactions. And 2 of them were someone's like master's thesis that I don't think went anywhere after that.
Yeah.
So as far as the mycoremediation approach, it hasn't gotten that far. There's a lot of exciting work going into the bacterial side of things. But once again, as far as using a bioremediation approach, I think that's far off, simply because the bacteria that do this sort of work are kind of oddball bacteria in that they have fairly specific growth requirements and they only like certain environments. And so they're not going to be, and they tend to grow really, really slowly, which makes sense since PFAS is not a good food. And so if you're trying to eat PFAS, you're not getting a lot of nutrition, you're not going to grow very quickly. And there's another issue is if you're trying to treat soils with a microbe, soil is so heterogeneous. There's so much other stuff out there. Most microbes, if you present them with almost anything else other than PFAS, they'll probably eat that other thing. Out in the real world, there's a lot of other things. And so for a bacteria to actually do that job in the environment for any length of time is probably not going to happen, which is one of the reasons I think the fungal co-metabolism is such a strength of fungi, because they don't need to tie PFAS degradation to them getting a meal. They can release their enzymes extracellularly, break down, like the lignocellulose, the other substrates they are eating, and sort of accidentally assault these PFAS compounds, whereas a bacteria pretty much has to be geared specifically towards it, and that's just really hard to achieve. But there's a lot of work going in water, like filtration and chemical and thermal treatments. The issue is all of our water treatment strategies can't be applied to soil simply because it's a solid substrate. You can't treat a whole agricultural field or a whole forest or something, right? If you're just thinking in just pure economics, if you're doing a chemical treatment, you can't afford like the 10 million gallons of whatever you need. Or if you're going to use a thermal treatment, it takes way more energy to heat that soil to high enough temperatures to break down the PFAS in there than it does to treat the water, because there's so much of it. And also the end product of all these treatments it's destructive. If it can destroy PFAS, it's going to destroy everything else. And so while you can treat the water and you end up with water at the end, or steam if you're using thermal treatment, which can then be reclaimed as pure water, if you do that in soil, you're going to end up with dead earth. And so that's not also a viable solution either. So just the infrastructure requirements, the energy requirements, and just like the pure mass, if you're using some sort of chemical compound to be introduced to the environment, are just at the get-go entirely uneconomical. And then you also have a host of downstream problems, like if you dump a bunch of chemicals into the environment to get rid of the PFAS, now you've got a bunch of chemicals in the environment. So it's nasty. And that's why we really need a soil treatment, and why some sort of biological approach is going to be something you'd like to use, because theoretically a bioremediation approach can be cheap and easy to apply. And it's going to need to be cheap and it's going to need to be easy if you're going to treat like the terrestrial environment, because you can't siphon off all of that polluted earth to like a water treatment plant. And then you just have to treat like a tank of water. You can't cart off the surface soil or the soil all the way down to the groundwater and try to treat all of it, right? I mean, people actually do that. That's how we treat the most contaminated areas, like Superfund sites. We literally just scoop up all of the soil and cart it off somewhere. And you just can't do that to the extent that's going to be needed to tackle this problem.
Wow.
Thank you.
Thank you both so much. This is just eye-opening and fascinating, and that makes me really proud for MMS to be supporting, even in a small way, your guys' research.